Hello, my name is Quinn Cavanaugh, and I'm an intern at the National Cancer Institute where I do research in the RNA biology lab. I've been invited here to share with you some of the do's and don'ts of molecular biology protocols. Today, we'll be detailing PCR purification and extraction, so let's get right to it. In molecular biology, PCR, or polymerase chain reaction, is a commonplace protocol used in labs all over the world. Since its conception in 1982, the applications for PCR technology have increased exponentially. Today, we use it for things such as genetic cloning, mutation detection, and even DNA sequencing. Today, we will be amplifying and extracting a fragment of microRNA from this plasmid. To begin amplifying, we need to create a solution that the reactions can actually take place in. There are 6 components to a PCR. First, we start with the DNA that we're trying to amplify, which can be from the genome or from another plasmid. Next, we have the primer, which is a synthesized strand of nucleotides that designates exactly what part of the DNA we're amplifying. There must be a forward and a reverse primer. The Taq enzyme uses these primers to recognize specific restriction sites, and then begins to anneal nucleotides according to the complementary strain. Finally, we need to add concentrated reaction buffer to create an environment that will be optimal for the Taq enzyme. We then add water to dilute the reaction so that we have a larger volume to work with later on. If you're adjusting to 25 microliters, you can use this table, which will show you exactly how much of each material you need per given amount of reaction. Just remember to always make enough solution for 2 extra reactions. One of these reactions will be for your control, or the reaction that has no DNA in it. The extra 25 microliters is just to ensure that you have enough solution in case you make a mistake. In a 1.7 microcentrifuge tube, add your water, nucleotides, and reaction buffer. The reason that you're combining these 3 materials together right now is because they'll be the same regardless of what DNA you use in the experiment. However, if you're trying to amplify multiple samples, make sure that you're using the respective primer to each sample of DNA, or else the region that you will amplify won't be the correct one. After making this delineation, add each of the DNA samples and their respective primers into each individual PCR tube. Finally, add the Taq enzyme to the main mixture and distribute 23 microliters of the solution to each one of the reactions. This includes the blank.
It's best practice to make sure that all of your PCR solution is combined. So before putting it in the thermocycler, make sure you centrifuge it.
Now that we have our solutions, we'll put each reaction tube into a thermocycler, which will allow us to input specific temperatures which correspond to each part of the PCR reaction. There are usually 3 steps to the reaction, starting with denaturing at around 95 degrees Celsius. Denaturing splits our DNA sample in half, leaving 2 strains. The temperature is then decreased to around 68 degrees to initiate annealing. This temperature allows the primers to bind to their unique complementary positions on the 2 strains, one reverse and one forward. The Taq enzyme then identifies these prime target sites and begins to move down the line in a process called elongation. Typically, this takes place at 72 degrees, and it allows the enzyme to bond nucleotides in the existing strain to its complementary nucleotide in solution. This process continues several times to increase the concentration of DNA, enough to where we can extract it. Now that the reaction is complete and we have our solutions, the next step is to actually extract the fragment that we amplified. We'll do this through a process called gel electrophoresis. First, we must determine how much agarose we're going to be using in our gel. It is essential that we correlate the concentration of the agarose to the insert length of the fragment. This is because gel electrophoresis separates samples by their sizes. Therefore, smaller sizes will run farther to the bottom because they are less dense, while all larger ones will stay at the top. For this insert, which is around 200 base pairs, we're going to be using a 2% gel. Since we will only be extracting a few samples, we will use this small cassette, which should only need about 50 milliliters of gel solution. To get our cassette ready for the gel, we must tape both sides and then place our comb in the top. Make sure to tape it like this, leaving a little bit of tape over each edge so that it will stick to the bottom of the other side, ensuring that nothing leaks out while you are waiting for your agarose gel to solidify. For this 2% gel, we'll need 1 gram of agarose and 50 milliliters of TAE1X buffer. Next, you must add the agarose and the TAE together in a flask, and then put it in the microwave for 40 seconds, swirl it, and then put it in for another 10 seconds, or until you can no longer see the crystals in the solution. Next, you must add Cybersafe, which is a DNA gel stain. It binds to the DNA and creates a complex that will absorb blue light, which allows us to image it with UV wavelengths. However, this gel stain will denature at high temperatures. So make sure that you can touch the bottom of your flask safely and comfortably before adding any Cybersafe to your agarose solution. It is optimal to have a 10,000 to 1 ratio of agarose to Cybersafe. So in this instance, for 50 milliliters of the agarose solution, add 5 microliters of the Cybersafe. Now that we have our solution, we're ready to pour our gel. Make sure that there are not any air bubbles around the comb or in the center of the gel. If there are, you can use a pipette tip to slowly drag them to the bottom left corner and either pop them or consolidate them so that they won't give you any issues in the future. Once the gel is solidified, we'll be able to load it with our PCR reactions. However, this may take a while for it to dry, so it is always advantageous to prepare your gel before your PCR reaction. In order to be seen in our ChemiDoc image, we need to add gel loading buffer to each of our samples. In this case, we will use Gel Pilot 5X loading dye. Add 1 volume of gel dye to 4 volumes of the sample. So in this case, it would be 6.25 microliters for every 25 microliter reaction. Now that our gel is settled and our reactions are ready, we can now start the gel electrophoresis process. To be able to interpret our results, we need the addition of a DNA ladder in our gel. This will definitively show us the length of each DNA band that is released. Depending on the concentration of your ladder, add 5 to 15 microliters of it into the leftmost reservoir in your gel. In the same fashion, add the rest of your samples to the subsequent reservoirs. In cases where we are trying to extract the fragment, make sure that you use all of your samples so that you can maximize the DNA concentration in the final product. Once we have loaded our gel, we can close the gel cage and attach it to a power supply. Typically, 160 volts is an appropriate level to get the gel to run completely through in about 20 minutes. Make sure that you see bubbles coming up from the side of the cage to ensure that the gel electrophoresis is actually active. Now that our gel is done running, we can use the ChemiDoc machine to verify the success of our PCR reactions. For imaging nucleic acids, we will use this blue tray. This machine uses UV light, which we can use to illuminate the different DNA bands and then compare them to the ladder. When we can compare it to the ladder, we can see the true size of the DNA band and see if it's the one that we desire. As we can clearly see according to our ladder, we have bands that were released at around 200 base pairs, which is what we desire. If we can go over to the blank and positively identify that there was nothing copied over there, then we have successfully replicated and amplified this fragment of DNA. Now we will cut the band out of the gel. To do this, we will place the gel on top of this UV light, which will cause the DNA fragments to emit green light, allowing us to see and cut the band. Make sure that you have protective orange eyewear to ensure that your eye isn't damaged by the harmful UV rays. Now, cut the band that we identified as the correct one in the image with your razor blade to isolate it. Now that we have our cut gel, we have successfully amplified and extracted a piece of DNA. However, the job isn't done here. To fully purify a DNA fragment, you will need to use some kind of gel purification kit. When it comes to purifying gel fragments, there are absolutely tons of protocols out there that you can use. The one that we use here at NCI is the Kaya Quick Gel Extraction Kit. Each kit has its own protocol that will walk you through step by step on how to purify your fragment of DNA to use it in an experiment. However, I will not be covering it in this video. Today, you have learned how to use the near universal protocol of PCR amplification and extraction. I'm Quinn Cavanaugh from NCI. Thanks for watching.